Theoretical genetics. Let's talk about that. How you figure out what traits you're going to get. All right? Probably don't need any iPads out. We don't have anything rolling on our iPads. We're just watching Mr. Hey, I'm going to be your show today. Just going to be me. Going to be talking. Going to be doing some things. Doing some Punnett squares. But I always have to bring into the fact that when we first moved into this building, I had a student in one of my biology classes. I asked him who the father of modern genetics was. And the answer was corn. No, the answer is not corn. It is, that's right, say it, Gregor Mendel. All right? Every time I hear Gregor, I think of, Meet the Parents when he meets, Luke Wilson, the ex-boyfriend. Oh, Gregor, look good, Gregor. I bet you're hungry, Gregor, when he's going into the... No?
I got you.
Okay. All right. These you have defined before. All right. Genotype. So let me go ahead and just give you some very easy ways to remember it. On the right side, you can write your any extra notes that you have. All right. Genotype, what I always say is G, genetic makeup. All right, kind of what a lot of the girls wear every day. Genetic, oh no, excuse me, not that kind of makeup. But what it is actually the letters that you're doing. A, B, B, C, C. All right, the genetic makeup, the actual genes. These are the genes that we're talking about. The genetic makeup. Phenotype is, and whatever the definition is, I look at it like this. Physical appearance. Should be easy to do after you did your homework. So the physical appearance. Brown hair. All right. Blue eyes. All right. Green skin. Whatever that may be. All right. Phenotype, physical appearance. Genotype, genetic makeup. Genotype means the letters. Phenotype, what it actually physically looks like. A dominant allele. All right, well, you have a big definition. What does it technically mean? The big letter, the capital letter, all right, is the dominant allele. That will mask every other recessive trait. So if you have a big letter, that's the trait you're going to be. The big letter. That's the phenotype or the trait you're going to be. Recessive, if I did this, A, B, C, D, E, F. All right, so my lowercase. All right, so the lowercase letters. The only way you see a recessive trait is if you have, what? Two of them that are recessive. So the only way that this is seen if they're in the homozygous state. So you have to have F, F, E, E, D, D, etc. Codominant, pair of alleles that both affect the phenotype when present in the heterozygous state. So that would be an AB, a BC, a DE, whatever you want to say. They're both going to kind of come together and have codominant. So two dominants, but two different genes that are affecting the phenotype. Locus, a particular position on homologous chromosomes of a gene. Homozygous, homo means what? Same. So I can have two identical alleles. I can have it both in the dominant state and the recessive trait. So this could be AA or a little a. Those are both homozygous, right? Because they're the same. Heterozygous means different. Hetero means different, having two different alleles, big A, or big B, little b. Okay? Carrier, individual that has a recessive allele of a gene that does not affect their phenotype. What does that mean? What is a carrier? What state do they need to be in that you've seen here? What state do they need to be in? Yes? Heterozygous. So if I'm this and this, here is the carrier. The person is not expressing that gene. Why? Because this is masking and dominant to that little gene. And then test cross, the stuff you guys were doing, Punnett squares and so forth. Okay. All right, now I got a good work on that. Oh, Gregor Mendel, father of genetics. All right, he had a couple babies. No, not really. He was a monk. Father. Sorry. All right, nothing. I got nothing. Okay, he was a monk. All right. Try to understand the principle of heredity studying pea plants. He looked at 7 traits. These are the traits that he looked at. Flower color. Well, we had purple and white. All right. So what do you think? What do you think he would have classified purple and white? Which do you think the dominant? Which do you think the recessive was? All right. So we get a big P. Now this is where you might get confused. What do you think letter would go with this? Little p. All right. Purple, white would be the little p. Axial and terminal, flower position. So again, the dominants are going to be listed on the left. The recessives are on the right. So these are all dominants. Dominant here and recessive here. All right, so those are dominant and recessive. Good news is you don't have to do any dihybrid crosses. That's the good news. All right. Prior to Mendel, the accepted theory of the heredity was called the blending theory. Basically, that offspring of the 2 parents possess a blend of traits. Not necessarily true at all. If somebody has brown eyes and somebody marries or has babies with a blue eye, you're not going to get a cross between blue and brown eyes and have half blue, half brown, or a mix of color between. Mendel's experiment contradicted this theory. When crossing a purple plant with white, he ended up with the offspring that were either purple or white. Conclusion, bless you, in his crosses, each trait had a dominant factor which masked or dominated the recessive trait. Everything that we know. No rocket science there. All right, some more important terms. The P generation is the parental generation, the first section that you are mating. We use this technology all the time in grape manufacturing in order to make new types of wine. This is just a classic way that we use this today all the time. All right, F1 generation, the offspring of a cross between the P generation. F2, when you take 2 F1s. So let me just go ahead and just draw this out for you so you understand what we're talking about. All right, here's mom. Here is dad. All right, there's dad, there's mom. Okay, parental generations. Go ahead and jot it down so you know when you're going back and looking at it what it is. All right. So what I'm going to do is F1 generation is going to be the offspring of a cross from the P generation. So here we go. I'm going to make this. Here we go. All right, here's mom. And here's dad. So you have room on the right to do it. I'm just doing it over the top. All right, I'm going to cross these. All right. So all I'm doing is bringing these down and putting them in these boxes. All right, A with one of these A's. Okay, so right now my F1 generation, what are they going to be? 100% what? 100% big A, little a. Here are your F1 generation. This is F1.
They're all
100%. No, you can't be anything else. So now I'm going to pull 2 out to cross. And I can pull any 2 out. They're all the same. So I'm going to pull this one and this one. All right, so let me do it right here. All right, so this is the F2. It's going to be in the middle. All right, so I pull these out. I got a big A and a little a, and I got a big A and a little a. All right, what am I talking about when I say big A, little a? What am I talking about? Oh, I'm talking about those, but what am I talking about when I just look at this?
Mom and dad.
What do we call? What's the term for this? This overall? Genotype. Genotype. All right, so now take a look how I do this. It's going to be very simple. I'm going to take this A and bring it here. I'm going to take this A and bring it here. Do the same thing. I always list the dominant one first. You don't have to, but it is technically correct. Big A, little a. This A is going to come down here with this little a, and then I got A, A. This right here is what generation? This is the F2 generation. And we have what? We have AA. We have A, little a, and we have little a, little a. And then all you do to figure out what the ratios are is write how many you have. How many of these do I have?
1.
So I write 1. I put my semicolon because it's going to be a ratio. How many big A, little a's do I have? 2. And how many little a's do I have? So what's my ratio? My genotypic ratio is going to be 1 to 2 to 1. Genotypic ratio. Easy or hard?
Easy.
Okay. Right. Well, you're going to get some time to adjust it. All right, but it's making a little more sense. Okay. So F1, the parents, F1, F2. If I was to ask you what the phenotypic ratio would be of this, phenotypic ratio. What do you think? I didn't tell you what A was. Let's say A was A equals purple color. I don't care what it is. Purple color. So what would be the phenotypic ratio of this? Yeah?
3 big A, 1.
All right, you just tell me what the color is.
3
All right, and then little a is, say white. Okay, that's fine. All right, white. So what would it be here?
3 purple.
All right, so, yep, you're good. So what color is this one going to be?
Purple.
Perp. Perp. What color is this going to be?
Purple.
Perp. What's this going to be?
White.
All right, so all you're going to do is add those up. 1 and 2, 3 perp, 1 white. So when you do phenotypic ratio, it's going to be a little bit different than a genotypic ratio. Pretty simple. It's making sense. Now, going on that question, what am I going to have for the F1 generation? What's my phenotypic ratio going to be? Yeah, 100% purple. You got it. All right? Or 1-to-1 purple, whatever you want to say. Okay, true breeding or purebred individuals that are homozygous for a particular trait. Big T, little t, little t. So that's true breeding or purebred. All right? Hybrid, heterozygous, cross-fertilization or fusion of 2 gametes by 2 different individuals. And self-fertilization, fusion of gametes by the same individual. Who does self-fertilization? Anybody in here? Anybody in here self-fertilize? No? You can't. All right, so what would do self-fertilization? Yeah, any type of plants. What do you do when you pull out those weeds and you blow? They're going to fertilize another plant with the little stuff that you blow off. Cross-fertilization can be done by you. It can also be done by plants. It can, you can have cross-fertilization of anything. But that is, some individuals can only do cross, some can only do self. All right? So just make sure you make a note of that. All right. Parental generation, we just kind of went over this, basically what I was talking about. There's a picture of what I just went over, right? Purple, white. All purple. Purple, 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 white. That matches up exactly. So draw an arrow linking what we just went over to that picture. And maybe it'll make a little sense. All right. Predicting the outcome of a genetic cross. Punnett squares are used in aid in predicting the probability of certain traits that will be inherited by the offspring. Probability, likelihood that you're going to get that trait. All right. Monohybrid cross, looking at only one trait. Dihybrid would be 2 traits. Trihybrid would be 3 traits. All right. That would be very complicated. A trihybrid cross would have 64 possible outcomes. A dihybrid would have 16. Monohybrid would have 4. 4, 16, 64. What would come after that if you did quarter? What? 64 times 4, right. 256, right. Hey, good. All right. So determine the dominant recessive trait. Assign letters for the trait. Use the genome type. And all right, so you're good. Complete dominance. Heterozygote has a dominant phenotype. Example: brown eyes is dominant to humans. Let's do the alleles real quick. All right. So what could I have for brown eyes is dominant to? Okay, so you have a big B and then what? Little b or? Big B, big B. Okay, so you got that. Write the 3 possible genotypes with corresponding phenotypes. All right, so let's say we have brown eyes, blue eyes. Okay, so we have that. What are the possible genotypes? We did that again. Here we go. Big B, little b, big B, big B. And if I have blue eyes, blue would be what? Oh, you guys are so good. All right, phenotypes, brown or blue eyes. Label which genotypes are homozygous and heterozygous. What are these? Oops, this should be a big B, sorry. Is this heterozygous or homozygous? Okay, I think we're good. Matt? All right, sample problem. You're going to try this one. In plant seeds, do it on the right. Round seeds are dominant to wrinkled seeds. Identify the ratios of genotypes and phenotypes in a genetic cross of 2 plants that are heterozygous for seed shape. And then what I want you to do is tell me the F1 generation. F1 generation ratios. All right, tell me the F1 generation ratios. I'll be walking around meandering. Grass is already done. I love it. Now just do the ratios. Tell me if they're round or wrinkled. Come on, Bruce. You already have it drawn. I already have it drawn. Let's go. Let's go. Let's go. Let's go. Ah, Margaret is killing it. Freeze Man straight bringing it. Allie Rock doing it. Well, now you got to have the parents are big R, little r. The parents are big R, little r. All right, so the P generation. So you know R, 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 R. So those are where you're crossing. So what do you have to do? You have to just make sure you know this is going to go here, this is going to go here, here, and right there. And then you kind of combine them together. That's better. Now you have to do the ratios, ratios. Do the ratios! The ratios, we just went over all the ratios. Okay. I'm ready. You should be ready too. Big R, little r, big R, little r. I'm just filling in what I see. All right, if you got that, give yourself a clap. A clap. That's only in victories and wrestling. Yeah. No. All right, now, somebody tell me what the F1 generation genotypic ratio is.
1 R, and then 2 big R, little r, and then 1 little r, little r.
Killing it, straight killing it. Allie Brock. Allie Brock, what's the phenotypic ratio?
3 round and 1 wrinkle.
Yes, yes. Oh, I know. Aren't you feeling this? Isn't this good? You want a quiz that's worth a lot of points? Okay.
Yeah, I'm down for that.
All right, try this one. Try this one.
Wait, so then what's the phenotypic ratio for the last one?
That's what she just said. Allie Rock said it.
What
She said 3 round, 1 wrinkled.
Oh, that's the. Oh.
Phenotypic. 3 round, 1 wrinkled. Do it. You can do it. Can anybody know it? Tell me the parental generation. P generation. P gen?
Got it.
What? Okay. Oh, go ahead, Kevin. Go ahead.
Uppercase B, lowercase b, lowercase b,
B?
Right, B.
Oh, sorry. Sorry. I thought you said E. I couldn't hear you. Let me try again. Hold on. Hold on.
I need the first one.
All right. Try again. I didn't hear you now. I got all confused. Try again.
Uppercase B, lowercase a, and then lowercase b, lowercase b.
Okay, there's a P generation. Woo-hoo-hoo. Man, I love biology now. Almost in the 3rd quarter, I get it all, Miss A. I get it all. Give me some biology. As long as it's not photosynthesis.
We done with that?
Photosynthesis, we're done until the final. Yes. But don't forget, why don't you a couple questions on final. Yeah, that's a lot. You already did the ratios here. Of course, do the ratios. Don't not do the ratios. Who's not going to do the ratios? All right, we're going to do a fire drill. We're going to give you 30 seconds. You know what a fire drill is? You get up, you go to another seat, and you check their answers. 1, 2, 3. If they didn't get it, leave them a note. Leave them a note. Go, go, go. 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 I want you to leave a good note if they got it right. Don't just stay at that desk for 1 second. Sit down. Stay at the desk wherever you're going. Sit down. We're doing a fire drill. I like the note. Alex is leaving a good note. Find it. Find one. Come on, sit. Noah, find a space to go. This is the easy part. Finding the empty seat. All right, here we go. Let's see if you got it right. So I'm going to take this. I'm going to make my little square. Yeah. I'm going to bring this right here. I'm going to bring this right here. I'm going to bring this over here. I'm going to bring this over here. All right, here we go. Big B, little b. Little b, little b. Little b, big B, little b. Little b, little b. Okay, now. So far, so good. So our genotypic ratio, Shickles, what is it?
2 uppercase B, lowercase b, and 2 lowercase b.
All right, or you could say 1 to 1 if you want. It doesn't matter. It's not incorrect, not wrong. Okay, and then phenotypic, Valerie, 2 black, 2 white, just like I like my cookies. The black and white cookie. There it is. All right. Good. If they did do it all correct, leave them a powerful note that makes them feel good for the rest of the day. And head back. Yes.
Does it matter if I wait, I'm gonna letter
Nope. Good day.
No, he ruined it. He said,
Ooh.
A plus
I have no
and then he
notes.
said such.
He said close.
Teddy. Teddy. All right, test cross. Now, this is the reverse. So if you have the F1 or F2 generation, you have to figure out what is the parent generation or what made those genes. Oh, I do. I know how to do this. I do. Right? To determine if a dominant phenotype is a homozygous or heterozygous. Cross with a homozygous recessive phenotype. This is the key to these problems. Right here, see this? Cross with a homozygous recessive phenotype. Example problem. Black is dominant to white in guinea pigs. You have a black guinea pig. How do you figure out if it's homozygous or heterozygous? What do you think? What would you do? All right, what am I going to test cross it with?
White.
All right, so black is out. You have a black guinea pig. So we don't know. So it's going to be this. So we're going to be big B question mark. All right, because that's black. All right, this is black. And then what are we going to cross it with? All right, little b, little b. All right, so then what we see is, what does the ratio end up being? So we got big B, little b. We got little b, we don't know. Little b, we don't know. And we got big B, little b. All right, so right now, not helping us. So we have a black. So we see the offspring that come out. Right now, what would we have? What would be the two offspring? We have two black and we don't know. So we would say, okay, well, if I did this and I made this a big B, I would have 100% what? If what? If F1 is all black. All right, you would have 50% white and 50% black if what? If black guinea pig right here, this guy right here, was what? Was what? Big B? Big B what? Little b. You understand? Let me show you what I mean. Okay, cross this off, make this a little b, little b, little b. Here's your 50% would be here. All right, so that's kind of how you fill in, you figure out which one it is. And what they're going to say is 50% were black, 50% were white. Was the mom or dad heterozygous or homozygous? Hetero. If all the offspring came out black, the black guinea pig was homozygous. Okay. All right, I think we get to do perform a test cross. So we can do this one. If you crossed a white guinea pig and got the following, what could you conclude about your guinea pig? Big B, little b, or big B, little b, perform a Punnett squares. So try this right here. Try these. Try to do those. You're going to have how many test crosses? How many? 3.
Does that mean you have to do 3 Punnett squares?
Yes. 3 big Punnett squares. I want to do A with you. I want to do A with you, and I'm going to leave you those 2. Check it out. So let's make sure we're working smartly. Check it out. I got half black, half white. All right, so I know that those 2 are what? Those 2 are white. Is it possible for me to have big B here? Big B here? No. So I would have to have this. And now all I have to do is go back and fill in. Well, this gave me a B, this gave me a B. Well, no, not that. What do you think? Right? So I filled in the Punnett square first based on this. All right? So, and by the way, you're crossing a white guinea pig. Right? You're crossing a white guinea pig. Oh, yeah, you're always going to have this little b on the left side, because that's the original one. All right, so try to do the next two. You're going to come around and check. Check, 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 check. Mic check, 1, 2, 1, 2. Check, 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 check. There you go. Really?
Check, check, 1, 2.
It's a weight.
I think we could do. I think we can do number. I think we can do D. I think D works. I think D works.
This one?
Yes.
It does. Next, if you have an all-white clean paper.
Yes, it works. I have it.
I just got it right now.
Yes, I have it.
That's exactly what I got. The reason it works. You see it?
Green is
impossible.
Yeah, if it's a
Is
white,
this right?
if it's a white department.
I think I got it.
Alex, how much?
You put the big B up there.
Correct one.
Where's the white one that you started with? This can't change, so keep that other one big. All right, so what are you feeling? I'm hearing some good answers. What do you think for B?
Impossible.
Not possible.
Wait, which one? B?
I kind of got it.
All right, why is it not possible? Well, let's say I have my white. There's my white, right? And, well, if we did big B and little b, let's see. Oh, well, that didn't work. All right, so let's cancel that. Let's do this. Watch. Whoop, whoop. Now what do we got? All black. So, not possible. If you got all black, what would be the other guinea pig? Big B. Big B, big B. This one was big B, little b, and that one was not possible. Okay. Good. Very good. Very good. We're rolling. We're almost done. You get to put pen and paper. Yes.
This packet is super.
You're still going?
There's just 4 more pages.
Yeah, we're going to roll here. All right. Incomplete dominance. So intermediate inheritance. Neither allele is dominant or recessive. That's kind of what I am, a mutt. All right, heterozygotes have a third phenotype that is a mix or blending in between 2 alleles. More and more of our society is becoming mutts. We're just blending all of our traits together. Right? Well, there's nothing wrong with it. Survival of the fittest. All right. Clue, if there are 3 phenotypes instead of 2, and one seems to be in between the other 2, alleles are written differently, no capital, no lowercase, usually a common letter with superscripts for 2 allele colors. Example, 4 o'clock flowers are going to be like pink, so you're not going to get anything in the middle of those. All right, here we go. The 4 o'clock flowers. All right, so let's figure out what our alleles are. All right, what are our alleles for this? All right, so example, the 4 o'clock flowers, color can be red, white, or pink. Write the alleles and possible genotype and phenotypes. All right, what do we have for alleles? What are the alleles going to be? Yeah, see? This is the allele. All right, here's the allele. All right, and they're all big C's. All right, so now, how do you know what the possible genotypes are? So what we do is we take a look. We have C, and like I said, here's the superscript, CR. CR. Or, what else could we have? CWW. All right. All right, or what else could we have? CR, CW. So you have a capital letter and like a superscript indicating that it's not a dominant or recessive. So it's not just one letter. You have the superscript on it. Very, very simple. What phenotypes could we have? Very simple. What is it going to be? Red, white, or this is how you get those pink flowers mostly. All right? Those pink flowers. Okay? So how do you get a pink? What is pink? Which one's pink? Okay, so this is pink. Just so you know, pink. All right? And the, obviously R would be red and W would be white. Yes?
Isn't that how you, isn't that, like mixing though?
It is like mixing, yeah.
Well, I thought you could do that.
No, you can, because now we're in incomplete dominance. You like the blending, you mean?
Oh, yeah.
Yeah. I mean, this is typical blending, yeah, for definition. All right, codominance. All right, codominance. A lot of you exhibit codominance in here. Both alleles are dominant, so express when together in a heterozygote. Alleles are written in the same way as incomplete dominance. Here's an example. A roan cow. It has brown and white fur. All right? Kind of looks red. Human blood type. We'll discuss that later. You have your A, your AB, your O's, your positive RH factors, your negative RH factors. All right. Codominance. All right. In Hereford cattle, coat can be red, white, or a roan color, caused by red and white hairs. All right, so what are our alleles going to be? We can just say, again, the same thing, C. All right, what would be the possible genotypes? CR, 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 CW, or, okay, I'll just put that one in the middle, and then CW, CW. What are the phenotypes? Red, white, and roan. Red, white, roan. All right, no different than incomplete dominance. Problem, cross a roan cow with a roan bull. Give phenotype and genotype ratio. So we're going to do this right here. It's no different than what we've been doing. We just add an extra letter to it. So what would the roan cow genotype be? CR, and you write CW. It's no different, you just add an extra C. And then CR, CW.
Does it matter, like, the fact that it's
Bull is male, cow is female.
like, I know, like,
Doesn't matter at all. Doesn't matter at all. So then we plug in, and what are we going to get? CR, 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 CW, CR, CW, CW, CW. What's that?
Oh.
Okay, so what do we get? What do we get?
You get
One red,
2 round, and 1 white.
2 roan, 1 white. And genotype ratio would be 1 CR, CR, 2 CR, CW, 1 CW, CW. Easy? No different? Is it any different? Say no. Good. All right, multiple alleles. Multiple alleles.
Wait,
Hold on.